Hey class, welcome back. Today we're going to start our next set of notes on Unit 6, Aquatic and Terrestrial Pollution, focusing on measuring the impacts of pollution. Primarily we'll be looking at how pollution leads to this process of eutrophication, which is one of the major impacts in aquatic pollution. We have bioaccumulation and biomagnification, again, in aquatic ecosystems. And then lethal dose 50 and dose response curves will be focused on as well in this section, talking about how we can measure these impacts of pollutants. To begin with, we'll look at a pretty extensive list, and you're going to want to pause this and fill out your notes in an organizer there, make sure it gets added in on your label it. Basically, there are different kind of categories of pollutants that we've talked about some of these throughout the year, others are kind of new. But essentially, we'll go through each row and talk about what they are. The pollutant, when we say there's a type of pollutant, it's excessive nutrients. Typically, these are nitrates and phosphates, so we can test directly for those things with kits. What are some effects? Algae blooms and eutrophication. Possible sources could be fertilizer runoff. That can be largely from agricultural sources, but maybe even domestic from your own yard. Organic wastes and feces, we can test with fecal coliform by doing a bacterial culture, looking for those, and also directly measuring the oxygen levels. The primary impact of organic waste is a decrease in dissolved oxygen levels, which can affect the fish and other organisms that live in this aquatic ecosystem. Not having access to oxygen is bad if you do cellular respiration. Sewage and agricultural runoff can be primary examples of this, so human domestic sewage waste and agricultural runoff from animal waste. The next type of pollutant could be sediments. We've talked a little bit about sediments. They can be tested directly, and these are sediments just like typically from runoff or erosion. They can be tested directly using something called a Secchi disc, and that basically is lowered into a body of water, and then we can see how long can we lower this before we lose a visual sight of it? And it's kind of black and white checkered disc there. Some impacts of sediments can be increased water temperature, reduced rates of photosynthesis as a result of the sunlight not being able to penetrate through the water as far as it would be able to if it were not cloudy from sediments. Logging, forestry, farming, construction, all these things that lead to increased erosion that we've talked about before can lead to increasing sediments. Salinity is the next one. We have total dissolved solids as how we can measure for those directly. That measures other things as well, but salinity can be directly measured that way. Stress to the organisms and disruption of food chain can be an issue. This can affect photosynthesis in plants as well. Road salts can be a big one, but anything that leads to increased runoff too can also be a problem. So the ones above kind of apply here. Heavy metal contamination. Lead, mercury, and arsenic are the three that we talk about most often. There are other ones, and we can test for those metals directly. Biomagnification is one issue we'll get into in a moment, and impact of human health, which we've already discussed in terms of the POPs and other impacts on human development. And then industries and mining, industrial sources of mining tend to be that. And then heat is thermal pollution, all right? So we can talk about thermal pollution, temperature changes that can affect oxygen levels. Warmer water tends to have lower dissolved oxygen than cooler water. Same with still water has lower dissolved oxygen than moving water. This can cause stress to organisms as temperature increases. We see this in industrial effluent outflow water. So something like an industrial source that uses water to cool something, or a power plant, the water heats up and then it's not polluted necessarily in any way, but it's warmer than the natural water and it's dumped back out into the main body of water, which changes the temperature. We can also measure indirectly for pollutants, so those were direct measurements. Indirect is indicator species. These are species that only live in areas that are unpolluted or polluted. So if we know about the organism's lifestyle, we can determine, something about that area in terms of its level of pollution. So as far as air pollution goes, which isn't the focus here yet, but lichens, remember there are fungus and algae kind of mutualistic organism. They don't thrive very well in areas with sulfur dioxide pollutants. So if we see a forest missing lichens entirely, that could be one indicator that there's issues there. Rat-tailed maggots are only found in polluted water, so we tend to see on our pollution level by these worms and maggots. When we talk about our macroinvertebrate studies, we really don't see them in high-quality water, so we have an idea about what's happening in the water supply if we see a lot of rat-tailed maggots. Gammarus are not found in high levels of salinity, so if we have really salty water that's supposed to be fresh, we would be missing this organism completely. We can measure the impacts of pollution by looking at dose response curves and determining the LD50. So when we study pollutants, we want to know how harmful something is. We can look at a dose response curve, and that really describes the effect that an organism, the effect of a pollutant on organism or mortality rates in populations based on particular doses of toxins in the environment, or sometimes even in drugs that are being studied. So in order to evaluate these dose response curves, we look for something known as the LD50, which is really the lethal dose of 50%. So that means the dose it's the dose of a chemical that is lethal to 50% of a population. This is one of those things where we study in a lab setting, and that lethal dose 50 is determined when half of the organisms that are being exposed to a particular dose would die. And that percent mortality is also referred to as the percent responding. We can study these in organisms in short term or long term, depending on what we want to accomplish. Acute toxicity is just how poisonous the substance is. Chronic studies are usually lower doses, but over a longer period of time to see how the chemical impacts these organisms over chronic exposure. Persistence is the length of time that chemicals are in the environment. So that's why we have table 17.2. Just talks about the half-life of different chemicals, and we've discussed half-life earlier. Some things hang out much longer than others, like DDT we discussed as a POP. It lasts 30 years in the soil. One term when we're looking at an LD50 graph is threshold. We need to understand the threshold is when we start to see response or death at a particular dose. So very low doses have no response, which means organisms can tolerate that. The threshold is when we first see an increase in response or death. The LD50 itself is marked at 50% mortality. Where did we find that on our particular concentration? And that's how we determine LD50 from that graph. We have impacts of pollution. So very specific impacts is eutrophication. This is a really big one. In eutrophication, we need to understand this occurs in bodies of water when we have really enriched nutrients. The increase in nutrients causes this big algae bloom, which you see in the background, this green high concentration of algae. What really happens is that the algae bloom grows from an increase in nutrients, so typically fertilizer, sewage waste gets into the waterways, so it tends to have runoff. The algae bloom grows, and this algae bloom affects the body of water in a couple ways. First, it directly blocks the sunlight from getting down to the bottom and supporting the ecosystem of all these plants here, and only has this massive bloom of algae. That causes some plants to die down here. It also, eventually, these algae will die and sink to the bottom. So you have that. So you get a lot of plant death here as the next step. The decaying plants and the death. Not only does the photosynthesis level decrease now because we've lost plants, but we're going to get these this dead, decaying plant matter to start to decompose by bacteria, and these bacteria are going to use a lot of the oxygen. So what ends up happening is the oxygen in the water decreases, and this lack of oxygen contributes to dead zones, and the fish suffocate and die off here, essentially. So this can be caused naturally. Sometimes this happens naturally from just nutrient erosion from our soil. But there's also this, if you ever hear cultural eutrophication, that's what we tend to talk about most often, is the addition of increasing nutrients, nitrogen, phosphorus from fertilizers typically. So what ends up happening in the impacts of pollution, we talk about this oxygen sag idea, is essentially this biochemical oxygen demand. It's the amount of oxygen required to support respiration by bacteria living in a water sample. So we have a couple of lines here. So we have types of organisms. We have a little bit of diversity here, which is good in these fish. And then the dissolved oxygen levels, the blue line, and biochemical oxygen demand is the red. You're going to want to mark this on your sheet, highlight it, or make different colors so you can see kind of how the oxygen sag curve looks, because we're going to graph this. So we have normal clean water. Here's our point source pollutant right here. So this factory dumping chemicals into the water. All right. So we have normal clean water. Now, at the site of pollution, we see dissolved oxygen start to decrease. Why is this happening? Because this is our decomposition zone. All right. Only pollution-tolerant organisms can thrive here, so just a couple of different species. And the oxygen starts to dip down, right? Because there's more decomposition. We have increasing bacterial respiration here that's sucking the oxygen out of this environment. The septic zone is kind of bottomed out. This is where just downstream, right? So the water's flowing this way. Just downstream, the pollutants are really high in concentration. Not a lot of organisms can survive here, and it's just kind of sludge worms, a lot of bacteria that are anaerobic, and the oxygen bottoms out, right? So dissolved oxygen continues to decrease. Biochemical oxygen demand is a demand of oxygen by these bacteria. So these bacteria are high right here, right? And then they start to decrease as we get this recovery zone further down the line as the pollutants are more diluted out and settled into the sediments. The bacteria then start to die off. Those bad bacteria, pollution-tolerant organisms start to recover and move in, and then ultimately the biochemical oxygen demand levels out at a very low rate where it's supposed to be, and the dissolved oxygen continues to increase into this clean zone once again. So this is our oxygen sag right there as a result of high concentration of pollutants creating this increase in oxygen demand by these bad bacteria, causes it to bottom out. And then as we move further down the line, we get back into a clean zone once again. The final type of impact we're discussing is biomagnification. This came up a little bit earlier in the year, but it's important to understand biomagnification versus bioaccumulation. Accumulation is just a buildup in individuals. So if you look here, you see these fish. We just have individual fish as they grow over their lifespan start to accumulate contaminants as they live and breathe, right? So toxins build up in individuals over time. Biomagnification is down here. Toxin concentrations are greatest at higher levels, trophic levels. So we have shrimp, small fish. We have our large marine mammals start to accumulate higher up. So higher up the food chain, the concentration of toxin is greater in those organism cells. Typically, these are fat-soluble types of things, so things like DDT, PCBs accumulate over a longer period of time, and they magnify through the food chain. So usually these are POPs. They persist for a long period of time. DDT was banned in 1972 as a pesticide that caused eggshell thinning in bald eagle populations as a side effect. It was a great pesticide, killed mosquitoes and other insects, but at the end of the day, its ability to magnify through the food chain was a really big issue for top predators like bald eagles. So ultimately, this is the per unit of body mass. That's the way to think about it here in terms of concentration being higher up on the food chain being affected. So, DDT, mercury, PCBs tend to be things that we see having the most nasty effects as they accumulate and magnify through the food chain. But ultimately, these things can have problems with humans too. So things like reproductive, nervous, and circulatory system issues as well.